a neuroimaging data scientist, and a biomedical researcher with a vast array of degrees and experience under her belt. After receiving her bachelor's degree in Cochin University of Science and Technology, she moved to do her master's and doctoral degree in biomedical engineering from New Jersey Institute of Technology and Stony Brook University, respectively. Currently working at the world-renowned Massachusetts Institute of Technology as a core member of the MRI team in the Brain Research Department, she's also working at the Ellen and Lorraine Bressler Clinical and Research Program for Autism Spectrum Disorders, Department of Psychiatry at Massachusetts General Hospital. With over 850 citations as a researcher, she's been an integral part of the world of translational neuroimaging. It's an honor to welcome Sheeba Arnold Anteraper to tell us about her work in functional neuroimaging as a tool for autism research.
Introduction. So it's an honor and privilege to be here at this monumental conference. The last time I was in Cerebellum was 22 years ago as part of a field trip to Sri Chitra. Some ways I'm so happy to come back and I thank Vijayaraghavan sir for this amazing opportunity. So I work at the McGovern Institute, and it was possible through philanthropy. So before I start, I have to really thank Suntech, UST Global and Trivandrum International School, because I believe donors are the lifeblood for anything, especially if it's a nonprofit. Okay. So as you know, advances in human brain mapping methods, be it beginning with lesions, through neuropsychology, through positron emission tomography, through DTI, all the way through the latest functional MRI, right? They have all gained new insights into human brain function, right? So today I'm going to focus on resting state fMRI. And we'll look into ways in which this modality can work its way through the clinic towards clinical translation. So when the conference began, Vijayaraghavan sir mentioned about the themes, inspiration, and today I'm going to keep like 3 themes for my talk, and it's personalized medicine, early intervention, and working towards novel interventions, right? So I'll start with the challenge in the field towards coming up with novel interventions. So if you PubMed for all the MRI studies that has been published in 2 decades, right? So you get about 20,000 hits. And if you work your way through the list and look at autism, in that period there was over 1,000 publications. And fMRI, as you know, is currently the workhorse of looking into human brain function in health and disease. But even though it has provided lots of insights into brain differences of neuropsychiatric disorders, this knowledge has not been fundamentally altered patient diagnosis or treatment. So the challenges are I see some MDs in the audience, as you well know. The etiology of most of these neuropsychiatric disorders are currently unknown, right? So there is this huge need to develop biomarkers. And the diagnosis is still based on symptoms. Often it comes late. Treatments are often based on trial and error, right? And in the case of autism specifically, right, there is a lot of heterogeneity. Someone mentioned if you meet a person with autism, you have met one person with autism. And for pharmacological interventions, they could often have side effects, and as you know, and it's only partially effective. So there is this huge push. Again, to reiterate the theme, there has to be some push towards personalized medicine, early detection, intervention, and also coming up with novel therapies and intervention. So as we work towards novel clinical translations, I believe that the items highlighted in green here can be accounted or in some ways addressed through functional MRI. So if you think of functional neuroimaging as a tool for autism research and explore the landscape a little bit, right? So in neuroimaging, you often see positron emission tomography and functional MRI, but in terms of publications, functional MRI is currently dominating the field, and within functional MRI, resting state MRI has taken over. And the reason for this is that resting state MRI doesn't require a task. So you can basically don't have to worry about ceiling effects, right, behavioral differences during task performance. You can put the subject, the child, inside the scanner, and the subject can just rest there. And then the resting state networks that come out, right, it can help you to come up with hopefully early identification, coming up with neuroprediction, and also personalized medicine. So I'm going to quickly explain for people who are new to this resting state network. It's this intrinsic architecture of your brain, and it occurs at this low frequency, and just to repeat, it's at the absence of a task. So you lie inside this MRI machine. Think of it as a brain camera, right? So you're lying inside the MRI scanner. You have nothing to do. You could keep your eyes closed or fixate on a crosshair or whatever. And these resting state networks just emerge. So you could look at it in seed-based ways or data-driven ways. So here in this example, I have a seed-based. So the black dots that you see, right, those are seeds. And these patterns that emerge are based on the temporal correlations or from the seed to the rest of the voxels or volume elements in your brain. Of course, the cerebellum here is missing in this picture, but I'll come to that later. You're only seeing the cortex here, but you could also gather a lot more from the cerebellum. So cerebellum is the structure that sits right under over here, which has a lot more neurons than the entire cortex combined. So yeah, I'll get to that a little bit later in the talk. And the applications, like I said, there's nothing to do, no task involved, so you could easily administer that to kids who are comorbid, who are on the lower end of the spectrum, and so forth. Okay, so there are many, many kinds of resting state networks. The one that's explored more is the default mode network. And then depending on where you place the seeds, different networks emerge. I'm just going to give one example. So this is from one person scan. I just scanned this person about 2 weeks ago. It's a 5-minute scan. There's nothing to do. And in 5 minutes, you get these beautiful networks. And default mode network, it's the most explored in autism because the patterns for these networks are very similar looking if you look at social processing regions. So, and social processing theory of mind kind of regions are kind of affected in autism. So it's easier if you explore the default mode network, and you can think of it as a functional tool to explore and understand this disorder. So it was discovered back in 2001. And typically in a functional MRI, you always, typically you have a task. And Marcus Reichel back in 2001, what he did was he, using positron emission tomography, flipped the question as to what are the regions in the brain that are more active when you're resting compared to when you're doing a task. And these networks just emerged, and it's beautiful because 14 years and 3,000 papers later, it's still one of the most studied network. And this is just a graphical representation. And the blue region that you see, that is all the papers reporting and citing this paper. And basically, I just put this in here just to give you a glimpse of how much relevant the default mode network is. And all of you have a default mode network, so it's very easy to explore. Okay, another good reason to look into default mode network is in the healthy brain, the greatest suppression of this network is associated with better memory formation. When the task gets harder, your default mode network is modulated, and it's also, in general, the better the default mode network is detached compared to the other networks. It's a proxy for your executive function. So, like I said before, the good thing about looking at default mode network is because of its overlap with social brain regions and social processing is a difficulty that's often implicated in autism. So, and if you look at a recent review, default mode network was the one that was highlighted, and that's looking atypical in autism. Okay, so in this iconic figure from this paper from 2005, what we are looking at in red is the default mode network, and what you're looking at in blue is the task positive network, which are basically working against each other. So, I mean, or anti-correlated, we call it, right? So if I pluck out a time course from this red region here, and plot it out, and if I pluck out a region from the blue region here and I plot it out, it looks like it's opposite to each other. And the more they are apart, the better your executive function is. So that's another reason to explore this network. Okay, so we did the study. So in A, you're looking at one study, and in B, the second panel, you're looking at another study. So the reason why we need multiple studies is that replication is a crisis in any clinical population. Clinicians would know this, that you might see something in one data set, but you need at least one or two more data sets to replicate it and make sure that you can be confident about your finding, right? So we did this a few years ago. It's published now. That in A, we had our cohort, and then in B, we had this cohort from this Autism Brain Imaging Data Exchange program that you can see, and it looks like it's more or less similar in ASD and healthy controls, but the biggest difference is that if you look at the blue patches, right? Like in the blue patches here, it looks very different in ASD. So that tells you something that the anti-correlation, correlation with the default mode network in ASD folks are slightly atypical or somewhat atypical and very atypical in many different regions. And that doesn't get us much closer to personalized medicine or neuroprediction or early intervention or anything like that. It just tells that the picture is slightly different, right? If you scan an autistic person and a typical person, of course, in spite of the heterogeneity, as a group, you see some differences, but what do we do with that, right? And the situation gets a little bit more complex if you look at some of the impairments. So during the panel discussion, someone mentioned interoceptive ability is affected. This whole afternoon session, there was this beautiful talk on sensory, how to help people with that, and then emotion processing. There was this all these apps and games, and so it's all there. But how do we come up a little bit closer? What would be a more complete picture? Was basically we wanted to get away from group results, and we just wanted to see how the data can lead us. So I'm going to give you some examples of data-driven approaches, and that's how our lab is heading towards. So stay away from group results, go towards like data-driven results, and head towards personalized medicine. So one example that I'm going to give today is again from a data sharing initiative. It's from the Yale School of Medicine. So before I go into results, I just want to give you a quick glimpse of what the power of connectome analysis. So every dot here is a region of interest, of course, the cerebellum is missing, and there's a reason to this, that back in the days, there's always a trade-off, right? So imagine like older cameras or older cell phones that when you take pictures, it looks grainier. So you need higher resolution. And if you still want to keep your higher resolution, you have to chop off certain regions of the brain. So in this picture, the cerebellum is missing because of that reason, that back in the days, you couldn't have everything. We still cannot have everything, but such is life, but we are getting closer, right? So just to reiterate what I just said, in imaging, I think it's regardless of fMRI, for any imaging, this is a problem, that if you want certain signal to noise ratio, and you still want very, very crispy, high-resolution image, again, think of MRI as a brain camera. Every step you go away from temporal resolution to gain better and better spatial resolution, you hit a sampling issue. The other is true that if you want to sample faster and then you go away from spatial resolution and you get a crummy looking picture. So what can we do about this? And recent technological advances in terms of coil technology or coming up with better and better software for gathering these pictures, such as simultaneous multi-slice images. This is getting, we can close the gap in that way from the technology side, that we could still have whole brain coverage, still cover the cerebellum here, which is this region down here. Right? You could still have whole brain coverage, still have a very good temporal resolution, and still have a reasonably good spatial resolution. So in this study that I want to show today, there were 59 subjects. It was downloaded from this Autism Brain Imaging Data Exchange dataset from Yale, and we had 35 people on the spectrum. The beauty of this data set is that your repetition time, for every 475 milliseconds, like half a second, you're getting a whole brain, and you could, if you scan for about 7 minutes or so, you could get close to 1,000 time points. So what we are going to next look at is like a flat map of the cerebellum. So if you look at me from here, and if you look at my cerebellum, and if you flatten it out, that's kind of how it looks like. So you just focus on the first panel for now. So this is a between group contrast. So the region that we are now looking at is all the regions in the brain. I'm not showing the cortex here, but still it's a whole brain study, that all the regions in the brain that are underactive in ASD compared to healthy controls. And that looks like the language network, and again, language impairments are fairly common in autism. And on the right two panels, what you're looking at is an emotion task activity from a human connectome project done on 1,000 subjects from a separate paper. So if you look at the overlap, it looks like what we are seeing is this language network just comes alive using a totally data-driven approach. I didn't go select one seed. It's just a whole brain approach. And we get these three regions, and how do you now interpret it, right? Luckily for us, there was another study that was going on in parallel, and we could, in a fairly confident way, say that the language regions and the other cognitive regions, like the social processing region and the emotion processing regions, are affected in autism. And it's like, okay, you can say that it's affected, but to show it with a picture with your own brain and to point that out, I think is a step closer towards coming up with interventions. Okay, so I want to show the resting state characterization. So what we can now do is, with data-driven approach, you come closer to this, and now you can see this yellow region and get this network, see this red region here and get this network, and then start to interpret it. So because it's now looking like a functional pathology, what it also gives insights to is into healthy brain in general, because you could look at how these networks look like in healthy controls, and then come up with what can be done to kind of help or restore these networks or along those lines. And that is not enough. I think we have to take a multimodal approach because resting state could still have challenges in spite of addressing like all these kids on the spectrum might move a little bit more while they are in the scanner and so forth. We still have to, I think, look at the structure differences. So this was the voxel-based morphometry study. So you look at every single volume element in the brain between healthy controls and autistic individuals. And that green patch there, that was the biggest difference that we were seeing. And it's interesting because each color represents a network. So this green is like the attention network, green and purple are the attention networks. So it's interesting that the attention network, and in the morning we heard about comorbidity, ADHD, is common in ASD. So for this makes a lot of sense. And if you could now seed that green patch and look for between group differences, and again, in light of the paper or the results we saw earlier, it makes a lot of sense that the regions that are typically anti-correlated now becomes correlated, and that might explain some of the self-rumination, more mind-wandering, things like that. That might explain some of the attentional problems. Again, this is still a group study. This is not an individual sprain that we are looking at. We are still looking at autism as a group and healthy controls as a group, and I have to point out that there's a lot of heterogeneity even within the controls, not just autism. Okay, so one other thing that we could do is we could do prediction analysis. So we could explore one data set and then put those in multiple data sets and see if the results are lining up. So we have started to do that with machine learning approaches. And one region that's consistently lighting up seems to be the cerebellum, particularly the non-motor regions of the cerebellum. For post-mortem findings, cerebellum is one of the most consistent regions, so it's making a lot of sense that even in spite of using like the latest sequences, the best available head coils, the best available data processing methods, that our results are landing on the cerebellum. Okay, so we can now zoom inside the cerebellum. So there is this region called the dentate nucleus. So we have started to do that because that particular region is one of the major output nuclei of the cerebellum. So the cerebellar cortex goes into this dentate region, back to the thalamus, to the cortex, and then it completes this loop. And because the basal ganglia is involved, and the comorbidity with Parkinson's and things like that are mentioned. So, I mean, later on in life, so it's an interesting region to look at, this dentate nucleus, and we have started to look into that as well. And we are seeing some differences. And that, not just differences, we could then start to look at behavior correlation. So each person with autism has a score. And we could start looking into correlations and so forth. So we see that our findings with whole brain data-driven approaches are helping towards the direction of coming up with biomarkers. This cerebrocerebellar disruptions, the cerebellum could serve as a target for therapeutic interventions and aid diagnosis and predictors of responses and help with managing the disorder. So we saw all of this, but I haven't spent much time on single subject imaging, and with higher field strength magnets, I think, and with higher resolution, I think that would be the direction to take. So we are looking at just one person's brain, again, looking at the default mode network, and then we could start doing this with better and better resolution. So this is like a very, it's like a sub-millimeter isotropic resolution. So the theme, I talked a little bit about early detection, looking at default mode network changes, and I kind of touched on the precision medicine, as you just saw, by scanning one person at a higher field strength, better resolution. What I haven't touched at all is coming up with novel interventions, so I think that's up to us as a group collectively. We can start thinking about once we are getting closer towards biomarkers, what can be done towards coming up with interventions along those lines, because autism is a neurodevelopmental disorder. So now that we have neuromarkers, how can we now try to change this network using interventions? So I know I showed a lot of brain pictures. For people who are not from neuroimaging background, it might look a little strange or whatever, but I just want to end with this quote from my favorite psychiatrist, Dr. Vikram Patel, that mental health is everyone's business, right? There is no health without mental health. So we have to have a lot of awareness, parent participation, and policy making in schools and so forth. So I want to thank, again, I would like to thank the organizers and thank my team, especially Susan Whitfield Gabrielli. Thank you.